So I'm going to try to record this today. We'll see how it goes. Okay, so the way that this kind of section of the class is set up is, since I don't have videos pre-made, my goal is to set up the lectures so that I talk for a little bit so y'all see some content, and then we break and work through some stuff on the activities. Okay, so I do have some copies of activities printed out. Hopefully some of y'all printed them out as well. But so the idea is that I'll be talking for 10 or 15 minutes, and then you'll get 10 or so minutes to work through a few questions, and then we'll kind of recycle that idea. Okay? So on Monday before the exam, we talked about nucleic acids, right? So really what we're doing here is we are going through the other classes of common biomolecules. So on Monday it was nucleic acids, and there were videos posted for those, so hopefully y'all saw them. They can help you finish up your activity. Today we're talking about lipids and biological membranes. We'll then move into carbohydrates, and then we'll kind of focus a lot more on some of the chemistry that happens in the cell catalyzed by enzymes. So we'll focus for several days then on how enzymes work, and talk about like some ideas of chemical kinetics and how that applies to enzymes and biological systems. Okay? So let's rock and roll. This part of class, I think, tends to be the kind of an easier part, lipids in general, because there's just not a lot of chemistry to them until you start talking about how they're synthesized and broken down. And that'll be later in the semester. Because really the structure of them is pretty simple. It's pretty straightforward. So we can, we can classify them lots of different ways. Okay, so there's really 5 main classes. There's fatty acids, which is the one that most people immediately think of, right? This is long carbon chains attached to carboxylic acid head group. Okay, it's always going to be a carboxylic acid on one end, and then a long fatty, and then a long carbon chain. Triacylglycerols. These are molecules derived from this molecule glycerol. Okay, glycerol is 3 carbons. Each carbon has a hydroxyl group attached to it. So the idea here is that on each one of these alcohols, there is a fatty acid attached via an ester linkage. Okay, phosphorylglycerols. The idea here is, instead of having 3 fatty acids attached to glycerol, you have 2 attached, and then on one of these ends, you have a phosphate, and then oftentimes another polar head group. Okay. Sphingolipids we won't really focus on. They tend to be really, really important in things like the myelin sheath, right, that wraps around neurons and protects neurons. But so they are just long carbon chains, and then instead of having a glycerol head group, glycerol, it's one of the alcohols on glycerol is substituted with an amine. Okay, so it's really just a substituted monoacyl glycerol. And then cholesterol, it's a really, really large class of molecules that are derived from this 4-ring system. Okay, so we'll look at 3 of these in a lot of depth, and then we'll talk about cholesterol a little bit. We're not really going to dive into the sphingolipids because at the end of the day, the chemistry is really, really similar to what we see with our triacylglycerols. Okay? And again, this is being recorded, so if you don't get all the notes down, it'll be online. Okay, so let's first talk about fatty acids. Can y'all see this okay? Do I should turn the lights down? Everybody good? Okay. So the general structure of fatty acids is pretty simple. It's a carboxylic acid group with a long carbon chain attached to it. That carbon chain can be saturated, meaning no double bonds, all single bonds, or unsaturated with at least 1 double bond. Okay, and we're going to talk about a little bit how important this double bond is, but the presence and the type of double bonds in these membranes, in these fatty acids, is incredibly important. Okay, so common lengths we'll talk about, but for bacteria, common lengths are 14 to 16 carbons long. For eukaryotes, it tends to be 16 to 18, with 20 also being relevant and important. Okay, and if you kind of just look at the general structure, right, it's as you expect. It's a long carbon chain. If they're saturated, they're all sp3 hybridized. If they're unsaturated, some of them are sp2 hybridized. But because of this, it ends up kind of looking like this zigzagging pattern, ultimately being kind of this straight line that zigzags within its parameters. Okay, and again, The most important thing is to remember there's always a carboxylic acid group on one end. Okay, if we think about the structure of saturated versus unsaturated, again, saturated, as hopefully everybody remembers from organic, means that all of the carbons are sp3 hybridized. Unsaturated means that there's at least one double bond. So there's at least two carbons that are sp2 hybridized. Okay, looking at this structure, right? So the saturated ones really do fit into that group. We talk about just this idea of kind of like a straight line with a zigzagging pattern falling into this line. What's really important to note about the unsaturated fatty acids is that they are, at least in the context of biological systems, those double bonds are always cis. That's important. The double bonds are always cis. If they're trans, that's called a trans fat, right? And that aggressively impacts the properties of the fatty acid, right? So if you look at the structure of this unsaturated fatty acid with the presence of this cis double bond, you very clearly see that it no longer is this straight line, right? So you have the straight line, a kink because of the sp2 hybridized carbons in the cis arrangement, and then another kind of straight line of carbons, right? So because of the double bond, we have this bend in our fatty acid chain. That's really relevant and really important when we start thinking about the structure of membranes. Okay, so the nomenclature. We could name these as chemists. We won't, because that tends to be a little more complicated, or we can name them as biochemists, because the naming convention is easier. And the reason, at least in the world of biology, that the naming convention is easier is because there's fewer things to worry about. There are themes in fatty acids in biological systems. Those themes are pretty simple, right? The really big theme is that when double bonds are present, they tend to be spaced by 3 carbons. Okay, they're always spaced out by 3 carbons, and they're always cis. So we don't have to worry about naming cis or trans. They are cis. If there's more than one double bond present, they're always going to be spaced by 2 carbons. Okay, or sorry, by 3 carbons, which means we can come up with a very simple convention to name these things. Okay, so here we look at some common names, and there may be some classes you take that require you to memorize the common names. I really don't care, right? There are some that you might recognize, like linoleic acid, alpha and gamma linoleic acid, arachidonic acid. You may have heard these terms before if you've taken like a nutrition class. But what it boils down to is this system right here. I don't know how well y'all can see this, but there's a number, a colon, another number, and then n minus something. Okay, so there's a number. We'll say 16, 0. What this means, the 16 colon 0, it means the carbon chain is 16 carbons long, and the 0 means 0 double bonds. Okay, so this fatty acid would have our carboxylic acid head group. This is carbon 1. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, right? The carboxylic acid counts as carbon number 1, and then 15 more carbons. This will always be the case if it's a saturated fatty acid. It'll be a number, almost always an even number because of the way these things are made. And then if the second number is a 0, that means it's saturated. There's no double bonds. If the second number happens to not be 0, happens to be 1, this means 16 carbons, 1 double bond, and because of the way that our biological fatty acids are arranged, we always know where it can be positioned based on one simple thing. So we write this n minus something. It can be n minus 3, n minus 7, n minus 9, n minus 6, n minus something. Let's just say n minus 6, okay? This means it's a 16 carbon fatty acid, 1 double bond. It is a biological fatty acid, so that double bond is a cis double bond. And the n minus 6 means the double bond is placed on the 6th carbon away from the omega end. And the omega end is always the carbon furthest away from the carboxylic acid. Right? If you go back to like your organic nomenclature, we think about, right, so this is the highest priority carbon. So alpha, beta, gamma, delta, etc. Right? But by naming this end always being the omega, it doesn't really matter how far away from our carboxylic acid we are. The omega end is always the very, very last carbon. Okay? So this would be considered an omega-6 fatty acid. The double bond is 6 carbons away from the omega end. So when you're drawing these unsaturated fatty acids, I think the easiest way to do it is to always start from that omega end. Okay? Otherwise, placing the double bond is going to be weird. Okay? So we start from the omega end, carbon 1, 2, 3, 4, 5, 6. This is carbon 6 right here. This is where the double bond is. For simplicity, I'm going to draw it like this, right? So 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Okay? So the first carbon of the double bond is the 6th carbon away from the omega end. So the double bond is between carbon 6 and 7. And I say for simplicity, I drew it like this just so that I can draw it as a straight line. Okay, but I want y'all to remember that when these are present as real biological molecules, they're not linear, right? So we draw it for convenience sake, but in reality, the presence of the double bond means that the chain is kinked. Okay? So this is 16, 1, N minus 6. It's a 16 carbon omega-6 fatty acid that has 1 double bond. If we switched it up and had 16, 2, N minus 6, okay, if it's 16, 2, N minus 6, then the same thing goes. It's 16 carbons. The first double bond is 6 carbons from the omega end, but the placement of the second double bond is inferred because double bonds are always spaced by 3 carbons. Okay? So I'm going to start from the omega end. 1, 2, 3, 4, 5, 6. This is carbon 6. The first double bond is between carbon 6 and 7. This is carbon 8. This is carbon 9. So this is the second double bond. 10, 11, 12, 13, 14, 15, 16. Okay? This pattern is always going to be real. In biological fatty acids, there is always one sp3 hybridized carbon between double bonds, which means the double bonds will always be spaced by 3 carbons. If we have an omega-6, we need to have 2 double bonds. The double bonds will be on carbon 6 and 9. If we happen to have a 16,3 N minus 6, it would be on 6, 9, and 12. Make sense? If this were 16, 2, N minus 3, the only thing that would be different is that this first double bond would, instead of being on carbon 6, would be on carbon 3. So we'd see a double bond right here. Okay, so if you understand how that works, then drawing these things and inferring the structure from a very simple name is very straightforward. Okay, any questions? All right. Okay, so then triisoglycerides. So this idea is pretty straightforward and simple. These molecules are derived from glycerol, and we have 3 fatty acids attached to the 3 carbons of glycerol, and they're attached through an ester linkage. Okay, so the way that these form is pretty straightforward. Right, so this is not surprising chemistry. It's straightforward carbonyl chemistry like we've seen before. I'm not going to bother drawing out all of glycerol or all of the fatty acids. I'm just going to kind of show generally how this works. So here we have the hydroxyl group of glycerol. Here we have the carboxylic acid group of a fatty acid. Straightforward. I'm not going to bother. Right, so generally speaking, we can imagine a base deprotonating this. But ultimately what really matters is that this alcohol is a nucleophile. It attacks the electrophilic carbon. I'm going to skip the intermediate because at this point I just want you to kind of understand generally how this works. We form an intermediate that collapses and hydroxide is lost. Okay? This is a condensation reaction, that ultimately produces water. The OH from the carboxylic acid and the proton from the alcohol end up being water, and we have a bond between the alcohol oxygen and the carboxylic acid carbon. So we end up with these ester linkages. This is called esterification. We esterify the fatty acid. It starts as a carboxylic acid, and we end up as an ester. Okay? Just some, I guess, other interesting tidbits. So these triacylglycerides, these are the basis for fat tissue, right? Our adipose tissue, it's packed triacylglycerides. Okay, and really the only difference between brown fat and white fat, if you've learned about this in your anatomy courses, right? Brown fat is lots and lots and lots of fat globules packed into adipose, whereas white fat is really just a single droplet in each cell, right? So that's really the only difference. Animal fat, our fat tends to be very saturated triacylglycerides, and hopefully this makes sense, right? So if they're saturated, they're linear and they can pack together better. But if we happen to have a lot of unsaturated fatty acids as part of our triacylglycerides, then because of those kinks in the cis double bonds, it would require a lot more space. So we wouldn't be able to pack as much energy into a single space. So by having saturated fatty acids as part of our triacylglycerides in our fat tissue, we can pack a lot more carbon into a smaller space. So they're a lot more energy dense. Seeds, these tend to be primarily unsaturated because they are a very, very useful form for immediate use by cells as they're trying to grow, or by plants as they're trying to grow. Okay. So ultimately, this is kind of what they end up looking like, right? So if this is our glycerol backbone, we have 3 fatty acids attached to each of our 3 carbons, right? So what those 3 are, really, there's no absolute requirement. If we're in fat tissue, it tends to be saturated. If we're not in fat tissue, it tends to not be saturated, right? So in this case, on carbon 3, we have an 18 carbon saturated fatty acid. In carbon 2, we have an 18 carbon unsaturated, right? So 1, 2, 3, 4, 5, 6. So this would be an 18 carbon omega 6 fatty acid. And this is a 16 carbon omega 1, 2, 3, 4, 5, 6, 7 fatty acid. Questions so far? Okay. So before we move on to, actually, let's do this real quick. So saponification, this is really chemical breakdown of fats, right? So if the way that, at least historically, we used to make soap, right, is you would take lard, right, from your animals, just lard, and add lye to it. Lye is just sodium hydroxide, right? Really, really caustic conditions. And under these conditions, under really basic conditions, you hydrolyze these ester bonds. And when you do, you release glycerol, right? So the ester bonds get hydrolyzed, you reform your alcohols and your carboxylic acids. But because we're under basic conditions, the carboxylic acids are deprotonated, okay? And that's how we make soap. Soap is really just fatty acids. That's it. So of course, the reason that it works this way is because of the fact that we have a hydrophilic and a hydrophobic portion, right? This is the same reason why biological membranes function the way that they do. So we have this soap molecule, and if you have lots of them around, really they can adopt 2 structures. One of the structures we'll see with the lipid bilayer, and nothing new to probably everybody in this class, like the lipid bilayer. The other form that fatty acids can adopt is a micelle, right? This is where we don't make a bilayer, but instead the fatty acids congregate by themselves and they form this sphere or this micelle. On the outside of the sphere is where we can interact with water. So our polar head group interacts with water, and on the inside it's nice and hydrophobic. So the idea is we can just capture a dirt particle or what other kind of like hydrophobic particle is in there. Maybe it's a virus, right? Trap it inside the micelle, and it's then sequestered from anything else. All right, phosphoglycerides. Really similar idea as triacylglycerides. you have a glycerol backbone. This glycerol backbone has 2 fatty acids attached to it. Okay, on carbon number 1 and carbon number 2, we have fatty acids. And then on carbon number 3, we have a phosphate and a polar head group, right? So the phosphate kind of forms the basis of our polar head group, and then quite often we'll have another molecule attached to that to make our polar head group. Okay, so this X group here, this can be H, right? So you just have a phosphate, or it can be lots of other things. You may hear things like phosphatidylethanolamine, phosphatidylcholine. These are common fatty acids. What they're talking about here is what kind of polar head group they are. So if it's phosphatidylethanolamine, that means we have a phosphate, that's the phosphatidyl part, and then the rest of the glycerol backbone. And then the ethanolamine, we have a CH2, CH2, NH3. Ethanol, amine. Phosphatidylcholine is 2 carbons, a nitrogen that has 3 more methyl groups around it, right? So phosphatidylcholine looks like this. It's your phosphate, 2 carbons, and then a tertiary nitrogen. So it's always got a positive charge. And there are lots of others. We won't go through them. Some are sugar-based, some are hexose-based. So there's a lot of different possibilities. But at the end of the day, these are the common ones. Okay? Now, the last really kind of big thing to understand about these is that they have very, very strict requirements. On carbon number 1, there's always a saturated fatty acid, always, no exceptions. On carbon number 2, there's always an unsaturated fatty acid, always, no exceptions. And on carbon 3 is where our phosphate group is, and then perhaps another molecule forming the polar head group. Okay, so it's cheesy, it's dumb, but I always remember a sup. Carbon 1 is it's always that pattern. And the reason for this is really important, right? These molecules form the basis of our lipid bilayers, okay? By having one saturated fatty acid and one unsaturated fatty acid, this means that these things allow the membrane to maintain some fluidity, okay? And we'll talk about that here shortly. So having the presence of at least one double bond means that these things can't line up to be super rigid. They have to have some space so the membrane can stay fluid. And the presence of the phosphate group and the polar head group on carbon 3 means that we are amphiphilic. So we have the ability to interact with hydrophilic regions and hydrophobic regions. Okay. The last thing I want to touch on before I let you start working on your activity is, I talked more than I thought, is that the placement of these double bonds is important, right? So the reason that, like from a nutritional perspective, that we've made a big deal out of like the idea of omega-3, omega-6 fatty acids is because as humans, as mammals, we can't make them. Okay, so as mammals, we can only put double bonds onto fatty acids 9 carbons away from the carboxylic acid. Right, so if we look at this fatty acid that we drew over here, here's the carboxylic acid. This is carbon 1, 2, 3, 4, 5, 6. This is carbon 7 away from the carboxylic acid. Our bodies, our cells, can make that double bond. This is carbon 8, carbon 9. This is carbon 10. Our bodies cannot make this double bond. We do not have the machinery to make a double bond to oxidize carbons that are more than 9 carbons away from the carboxylic acid. So any fatty acid you see that has a double bond 9 carbons from the carboxylic acid group, we can make, right? So if there's one between 9 and 10, we could do that. We cannot do any of them starting at carbon 10 or beyond. We have to get those in our diet. Okay? Those are called essential fatty acids, ones that we have to get in our diet. Any questions? All right, if not, we'll take about 10 minutes and you can work through the first bit of your activity. Anybody need them? Lots of people.